Hello everyone, welcome back. I'm going to go through how to do activity 4, 5, and 6. I'm going to really just breeze through this because there are great videos and resources for you guys to watch. I expect you guys to go ahead and watch those videos. So activity 4. So make sure you guys are following along in your lab manual here. So here we are going to be using DNA technology to perform an extraction. So whenever you get a specimen, you first have to extract the DNA from that specimen. So in this example, we are going to be using a strawberry, and we're going to extract the DNA from the strawberry. So remember, all living organisms are going to contain DNA. And you can extract DNA using everyday materials. Obviously, we wouldn't use these everyday materials to extract DNA in a laboratory because you want to make sure that your extracted nucleic acid is very pure. But for this purpose, it works well. So here's a video you guys can watch, and I also uploaded a video of a professor from RCC performing DNA extractions in her kitchen. So go ahead and watch that as well. So here are the steps. So first you're going to obtain strawberries. You're going to add, I believe they say 200 mils of cold water to make a strawberry soup. And then you're going to strain this strawberry soup. You're going to add soap and let it sit for 10 minutes. You're going to pour strawberry soap mixture into test tubes and add a pinch of meat tenderizer into each test tube. You're going to add isopropyl alcohol to the mixture, and then you will see your DNA. And there are specific reasons why we use these materials to extract DNA. So during the extraction, we try to keep everything cold. This is important because DNA and RNA are very sensitive. So here we give two main reasons to keep everything cold. So the first is that we use cold alcohol in order to cause the DNA to precipitate and making it more visible. The second is, like I said, DNA is very fragile, and there are various enzymes. For DNA, there are DNases, and for RNA, there are RNases that will degrade our DNA or RNA depending on what you're isolating. And these DNases are not active when the temperature is too low. So that's why we want to keep everything cold, because we want to prevent the degradation of our nucleic acid. So every time I've done extractions, I usually do RNA extractions. I've done DNA extractions a few times, but I typically do RNA extractions. And you, in the laboratory, you will use an enzyme called proteinase K. So in addition to keeping everything cold, you will also use proteinase K in order to make sure that these DNases and RNases cannot degrade your nucleic acids. So why do we use soap? Well, strawberry DNA is not the only molecule inside the nucleus. Strawberry DNA is not only inside the nucleus, but also inside the lipid bilayer. So soap is going to open the lipid bilayer and the nucleus to release the DNA. So soap or detergents are typically used in science to open up the lipid bilayer. This is not just specific to DNA or RNA extractions. We do this for a variety of techniques. We typically will use like a soapy detergent to open up the plasma membrane of tissue in order to stain it with different antibodies and look at it under a microscope. So this is a very common technique to use. And then why do we use meat tenderizer? So DNA has proteins that are associated with it. The meat tenderizer removes those proteins, making the DNA unwind and making it easier to see. So there's a lot of proteins that can be found on your DNA. And this tenderizer will help remove those proteins so that way we can really unwind our DNA and look at it a little easier. The other question in your lab manual is DNA polar or nonpolar? So DNA has nonpolar bases, which we discussed are nitrogenous bases, and those aren't found inside the double helix. However, DNA also has charged phosphate groups, which are polar, and these are found on the outside of the double helix. So that is one way your book describes extractions. Like I said, this is a very crude method. If you were in the laboratory, you use sophisticated methods to perform DNA and RNA extractions. These extractions, there's a lot of different ways you can perform these extractions. I'm just going to list a few. So for example, you could use Trisol reagent to cause these layers to separate. That's also a very crude method to do extractions. You can also do the spin centrifugation method. And that method, you also use isopropyl alcohol. That is a more common technique to use. And then there are more sophisticated methods which would use magnetic beads to perform extractions. And I think I posted videos of how to do it with the spin centrifugation method, because that's a very common method. It's used in research laboratories, it's used in government laboratories, commercial laboratories. So it's a widespread method. Okay, so let's move on to activity 5, which is PCR amplification and DNA fingerprinting. So DNA profiling is very important. It really transformed the fields of forensics because it kind of gave forensics a more scientific analysis of evidence for crime scene investigations and other legal proceedings. So here you can analyze DNA samples to determine whether they came from the same individual. And DNA profiling is also called DNA fingerprinting, especially when it comes to forensic science, because like your fingerprint, your DNA is unique to you and you alone. So for example, if you collected a swab from a crime scene from a blood, you collect a swab of this blood contains DNA. It also contains other molecules, but it contains DNA. Say you have various suspects, you have suspect 1 and suspect 2. You can do a buccal swab on suspect 1 and suspect 2. So buccal swabs are just getting like a cotton swab and swabbing the cheek of these individuals. So now you have a swab with blood sample, a buccal swab from suspect 1 and a buccal swab from suspect 2. Now you can isolate the DNA from these swabs. Like I said, these swabs are going to have other proteins and material present, but all you are interested is the nucleic acid. So you could either isolate the DNA, or what I'm more familiar doing is isolating the RNA. So you would use an extraction method to isolate the DNA. So like I said, there are various extraction methods available. And once you have your pure DNA or pure RNA, you want to amplify that. You want to make a lot of copies so that you can visualize it. So this is referred to as, so there are a lot of different techniques also available to amplify DNA or nucleic acids, but the best technique used is PCR. It's really the gold standard. So you would amplify this using PCR. And I don't really work with restriction enzyme digestion a lot, but you can also use restriction enzyme digestions in DNA fingerprinting. And then you will run it through a gel electrophoresis. So you will run it through your agarose gel, and your strands will separate based on size, and that way you can compare the DNA collected at the crime scene to the DNA from suspect 1 and the DNA from suspect 2. So this is the DNA you collected at the crime scene. This is the DNA from suspect 1. This is the DNA from suspect 2. So as you can see, there's a band here, and there's also a band here and here for both suspects. Okay, there's a band here in your reference DNA, and there's a band here for your suspect 2. A band here for your reference DNA, and it seems like there's bands on both of these. But all in all, you can see that the bands from suspect 2 match up with your reference DNA. So this links suspect 2 to the DNA found at the crime scene. Obviously, you have to still do further investigations, because what if suspect 2 lived in this home and their DNA just happened to be in the home? So now you have to find something that ties suspect 2 to the, like if this was the actual blood of this suspect, then you can tie him to the location of the crime scene as well as the time of the crime scene. But if you just collected something some DNA sample that happened to cross into the blood, then that just suggests that this suspect was at that location at some point in time. So I hope that makes sense. Okay, so PCR stands for polymerase chain reaction. And like I said, this is how we amplify our DNA. Okay, so amplification happens in 3 steps. Oh, actually, let me go back. So before you can amplify your DNA, you have to put your pure DNA with something that we refer to as a master mix. So your master mix will be comprised of various things. Some of those things will be dNTP, so your nucleotides, primers, your Taq polymerase, which we will discuss further. And then it'll have some buffers in it, sometimes like magnesium chloride, things like that to help the PCR occur. So you put your pure DNA with this master mix, and then essentially in PCR, 3 things are going to occur. You're going to denature, anneal, and extend. So in step 1, your DNA sequence is going to be heated up, and it's going to cause your double helix strands to separate. So now it separates. This is referred to as denaturation. And then in step 2, you're going to anneal. So like I said, you have your master mix that has primers. These primers are specific to whatever you are looking at. I do a lot of COVID testing, and the primers we use for COVID testing are, I think I actually have a thing to show you. Let's see if this is going to work. It's not. Okay. Oh, maybe it will. Oh. Okay. So hopefully I'm sharing the correct screen. But in COVID testing, we use primers for ORF1A, which is open reading frame 1A. We also use primers for the N1 and N2 gene. The N1 and N2 gene are going to code for nucleocapsid protein, which is found in your virus. So you will use primers that are specific to whatever it is you are trying to investigate. So in step 2, annealing, your thermocycler is going to cool down, and these primers are going to anneal to your template DNA. In step 3, which is extension, you're going to have your Taq polymerase or DNA polymerase come in with those nucleotides that you put in your master mix, and they are going to add to the 3' end of the primer and extend your DNA. And then in cycle 2, these are essentially going to just double exponentially through every cycle. Typically, you do anywhere from 30 to 40 cycles, sometimes even 45. It really depends on the protocol that you are using. Okay, so what do you need for PCR? I kind of already went over this, but you need your pure DNA sample, your pure nucleic acid. You need Taq polymerase, your enzyme, to allow elongation or extension to occur. You need your nucleotides, so this is usually like your dNTPs. In order to make new DNA, you need to have nucleotides, and then you need your primers. So your primers are short pieces of single-stranded DNA or RNA. They're usually 20 nucleotides in length, and they are specific to whatever you're trying to visualize. If you were doing, I don't know if this is going to confuse you, but there's different types of PCRs available. The PCR we're going to talk about today is it's going to be endpoint PCR, and this is a qualitative measure. It's just going to tell you whether that gene is present, yes or no, period. There's other types of PCRs called real-time PCR or quantitative qPCR. In this case, you will also need a fluorescent probe in your master mix. Okay, So this is once again just explaining how PCR works. You have denaturation, which occurs at high temperatures. Your strands will separate. You have annealing, where your primers will anneal to your template DNA strand. This occurs usually at a lower temperature. You have extension, where your polymerase and your DNTPs will be added to your primers. And then this is the result after one cycle. But as I said, as you go through further cycles, these will they will just multiply exponentially. And when you end up at the end of the 35 cycles, you're going to have so much DNA. So your DNA is going to be amplified from the original starting point. So here are some videos for you guys to see it in action, which I've already posted, and I hope you guys were all able to watch those. But okay, so we extracted our DNA from a sample. Now we need to, no, we extracted our DNA from a sample, and then we amplified it using PCR. Now we need to run it through a gel electrophoresis. So like I said, this is endpoint PCRs. So here you will make an agarose gel, which I posted a video how to make an agarose gel. It is connected to a apparatus, and this apparatus has a negative charge here where your wells are shown. These are your wells that are made using a comb, and this is your positive charge over here. So since DNA has a negative charge, you load your sample here, and when you turn on this apparatus, it will cause your samples to travel from the negative end to the positive terminus. These samples will separate based on size. So your small samples will be able to travel further compared to your large sample, which won't travel as far. We'll stop somewhere around here. And then your medium sample travels somewhere in the middle. So these DNA fragments are going to separate according to size. So in DNA fingerprinting, you can also use these restriction enzyme digestions. And essentially, these restriction enzymes are going to cut your DNA in very specific manner. The way these restriction enzymes will cut your DNA is going to be specific to the individual. And then you can run this through your gel as well. So here's your amplified crime scene DNA, and here's your amplified suspect's DNA. Okay. So this is just going further into how this gel electrophoresis works. So I hope you guys had time to watch the videos and read these PowerPoints. But these are some videos that I already posted for you to watch. So now I just want to check your understanding. So here on the left, well, let me set this up. So up on the top would be our negative charge. Over here would be our positive charge. This is our agarose gel. We have wells here. So this is our well, our well, our well, our well, our well, where we will pipette our sample into. So usually in your first well, you like to pipette your DNA ladder into it. This is your reference ladder. So all these strands have known sizes. So if you look at this strand, for example, this strand is known to have 500 base pairs. This strand is known to have 3,000 base pairs. As we've already discussed, the smaller DNA fragments will travel further. So 500 obviously is going to travel further than your 3,000. And then you will look at your other lanes. These are your, I guess, like you could say suspect 1, 2, 3, and 4. So which lane, 1 through 4, has the largest fragment of DNA? So in this case, 1 has the largest fragment of DNA. Which lane, 1 through 4, has the shortest DNA fragment? In this case, 2 has your shortest DNA fragment. Okay. So activity 6 would be using DNA technology to solve a crime. So you can read the scenario in your book, in your lab manual, but essentially you have some hair strands left behind at the crime scene, and the custodian wants to extract the DNA from these hair strands in order to determine which individual caused this mess in the um, life science department laboratory. So the first thing you want to do is extract your DNA from this hair sample. So assuming the DNA has been extracted from the sample, what procedure would you do next? Next, you want to amplify your DNA. So how can you amplify your DNA? You can use PCR. Once you've amplified your DNA using PCR, how can you visualize this DNA? You can visualize it using gel electrophoresis. So here we want to see who is responsible for the mess. This is the crime scene sample at the top. So remember, it goes from negative to positive. This is your well where you will pipette your sample into. And this is how your sample separated. These are the DNA fragments it separated into. These are all your suspects. So you have Dr. Thaler, Dr. Scott, Dr. Jazayeri, Dr. Johnson, Dr. Huff, and Dr. Taylor. Now you have to compare the DNA fragments from your suspects to the DNA fragments from your crime scene. And then, as I said, you always want to also load in a ladder as references for these, how many base pairs are found in these DNA fragments. So all these reference ladders, you know how many base pairs are found in here. So if we were to look at this, we see a DNA fragment here. Where else do we see a DNA fragment there? Dr. Johnson, Dr. Huff. So far, Dr. Johnson and Huff are looking like good suspects. So we have a DNA fragment here. We see in Dr. Johnson again, Dr. Taylor. DNA fragment here. We see in Dr. Johnson again. DNA fragment here. Once again, we see in Dr. Johnson. So who is responsible for the mess? It's safe to assume that it was Dr. Johnson based off of DNA evidence. So this is explaining endpoint PCR. I did post some videos describing how qualitative or real-time PCR are used. Like I said, endpoint PCR is just going to give you a yes or no answer. It's just going to tell you that this reference DNA matches this suspect's DNA, for example, in this scenario, but it won't tell you how much of this DNA matches this DNA. It's just answering a yes or no question. You're not getting a quantity from these experiments. You can get quantities from qPCR. So that is quantitative PCR. So I did post some videos there to help you understand how those can also be used in research. Okay, so that will be it for these activities. If you guys have any questions, please let me know.